Hey guys, welcome to PHU Biology. During the biochem video, I mentioned we were leaving out nucleic acids. So this video is going to be all about that group of molecules and DNA. There's a whole lot to cover for this topic, so it's going to need to be split into two videos. This video will cover everything about the structure of DNA, and we'll end with DNA replication. Next time I'll cover just transcription and translation. So let's begin. I want to start off by talking about the structure of DNA. So DNA is made up of these units called nucleotides that repeat themselves here, 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 and they repeat themselves to form chains of DNA. Nucleotides are going to be made up of three parts: a phosphate functional group, a ribose sugar, and a nitrogenous base. Hopefully you know functional groups, so this is a phosphate functional group over here, and it repeats itself here, and along every nucleotide there is a phosphate group. Next we're going to have a ribose sugar, which is this structure, and it, just like the biochem video when we learned about the structures of sugar, keep in mind that the corners of this shape are going to be carbons. And what may be new is the nitrogenous bases, these over here. So let's talk about those. First of all, do not worry about the structure of these bases. In DNA, we have four bases. We have adenine, we have guanine, we have cytosine, and we have thymine. Now keep in mind that RNA is also going to have four bases: adenine, guanine, cytosine, and instead of thymine, RNA is going to have something called uracil. Now, adenine always pairs to thymine with two hydrogen bonds. If we look back, adenine, thymine, two hydrogen bonds. And again down here, two hydrogen bonds. Guanine is always going to pair to cytosine with three hydrogen bonds. You can look back here. We have 1, 2, 3. 1, 2, 3. Now, what you do need to know about the chemical structure is this. Adenine and guanine are called purines, which means they have two rings fused together, C1, 2, and 1, 2. Now, thymine and cytosine are going to be called pyrimidines because they have one ring. Now, something interesting my biology teacher taught us to remember which bases fit into which category. The word pyrimidine has a Y in it, right? So because it has a Y, the bases with a Y, like thymine and cytosine, are going to be pyrimidines. And the bases without a Y are going to be purines, which also does not have a Y. So that's just a neat little way to remember. Alright, back to more general structure. When DNA forms, we end up with two polymers bound by hydrogen bonds. So we have two polymers, and the hydrogen bonds would be between the bases. And this forms the DNA strands. Now, DNA, when the strands come together, they form this shape that we call a double helix. Now, in the strands, the phosphate group is going to be at the top because this is a 5'. Whenever there's a 5', that means there is a phosphate group attached to the end, and that's simply because the phosphate group is attached to the number 5 carbon in the ribose sugar. So since the number 5 carbon is closest to the top, we call this the 5' end. Now if we look at the bottom, this is the 3' end, meaning that we have our number 3 carbon closest to the end of the chain. So that's how we denote the ends. Whichever carbon is closest would be 5 or 3. Now, we describe the strands as running anti-parallel to each other, because this strand runs 5' to 3', and this one, sorry, my pointer's frozen, there we go, this one runs 3 to 5'. So they run anti-parallel to each other. That's the word we use. Now let's go over some DNA replication. This happens in the S phase of the cell cycle. For a basic overview, DNA replication is going to be semi-conservative. This means that in any new DNA molecule, one strand is always going to come from the original DNA, and one is always going to be new. As you can see in this diagram, the old strand, the parent strand of DNA, is this bluish purplish color, and the daughter molecules each have one strand of this bluish purple and one strand of red, which is the new DNA. So that's how semi-conservative replication works. We're going to get a little more complicated and technical here, so bear with me. What we're looking at here in this picture is a section of DNA that's being replicated. We begin this process with an enzyme right here known as helicase. Helicase has the job of unzipping the DNA by breaking the hydrogen bonds between the nitrogen bases. So we get what we're looking at in this picture. This is actually called, this angle looking thing is called the replication fork. Now, along this chain of what's now open DNA, comes an enzyme called primase. You can see the primase right here. Now, the primase comes in and attaches to specific regions called primer sites. And at primer sites, it lays down an RNA primer. Since it lays down an RNA primer, remember that it's going to lay down complementary bases to what's here. So if here is adenine, it would lay down uracil because it's RNA. So keep that in mind. Now, this may get a little confusing, so just try to follow along with me. After primers comes an enzyme called DNA polymerase 3. You can see it here and you can see it here. DNA polymerase 3 is going to add complementary nucleotides of DNA instead of RNA. This time it's DNA. But this enzyme only works in the 3' to 5' direction. It can only go this way, along the DNA strand. So it can only work continuously and smoothly on this strand. See, because the green here is what it's adding to. This is the 3 and this is the 5. So it works smoothly and continuously here. So we call this the leading strand because it just goes continuously adding DNA. The other strand then is going to be called the lagging strand. On the lagging strand, DNA polymerase 3, since it needs to go 3' to 5', is going to go between several of these RNA primers, and it's going to lay down a series of fragments of DNA and then stop. Now, all these fragments, like this fragment and this fragment, they're called Okazaki fragments. So this takes longer than the leading strand. That's why it's called the lagging strand. After this has happened, so after DNA polymerase 3 has gone through both strands, we get another enzyme called DNA polymerase 1. You can see DNA polymerase 1 right here. DNA polymerase 1 converts the RNA primers that I told you about right here back to DNA, essentially just replacing the uracils with thymine. Then an enzyme called ligase right here joins all the DNA fragments together, and we end up with 2 new double helixes of DNA. So to sum it up, helicase unwinds, creating the replication fork. And that's this picture. Primase bonds to primer sites and puts down RNA primer right here. DNA polymerase 3 can only form strands traveling 3' to 5'. So on the leading strand, it forms a continuous chain, and on the lagging strand, it forms several Okazaki fragments. DNA polymerase 1 converts the RNA primers to DNA, and ligase bonds everything together. Hopefully the process is a little clearer now. Obviously, this is something that needs to be studied a lot and memorized. Well, that finishes our first lesson on DNA. If this video is helpful, leave a like. You can comment with any additional questions you may have, and don't forget to subscribe to see more biology videos. Thanks for watching.